My name is Dr. Andrew Heyman. I'm the Program Director of Integrative and Metabolic Medicine at the George Washington University. We have a very exciting upcoming conference on the relationship of the gastrointestinal tract, the immune system, and neurologic function. We have a number of experts that are joining us for this weekend, and I'm delighted to be interviewing today Dr. Eva Sapi. Dr. Eva Sapi has done some seminal research on the role of biofilm in chronic infections, which is a very important but not well understood phenomenon, and we'll be placing that into both a research and clinical context at our upcoming weekend. So thank you very much, Dr. Sapi, for joining us. As I said, I'm excited and delighted to have you involved in our teaching program and to join the faculty at George Washington University. So tell me a little bit about your research background and how you made your way into investigating biofilm.
Thank you very much, Andrew, for inviting me. And my research background actually is in cancer biology. I was a cancer biologist at Yale for more than 15 years. And I received a tenure-track position at the nearby university. And at the same time, I got Lyme disease very early. It was a very difficult time for me. And that is the first time I did not understand how we cannot cure something as simple as bacterial infection. So I actually changed my research interest, and I started to answer simple questions, why we cannot eliminate something like Borrelia spirochete. So the first couple of years, I just practically repeated some very known results. I used very standard antibiotics and tried to kill Borrelia in the lab, and I could not, and I didn't understand. So I at this point I was talking to some Lyme researchers, and they asked me to look under the microscope and see what's happening when you expose Borrelia to antibiotics. So we did, and I started to see something very, very interesting. When I gave antibiotics to the culture, I started to see some very interesting formations. And at first, I thought it's just maybe some side effect, maybe just some Borrelia is dying, and maybe just some waste product. But after I started to understand that those colonies, first I thought it's colonies or aggregates, they are growing. And instead of, I'm eliminating the bacteria, actually I'm created almost a little monster. So at that point, I was very, very interested in what's going on, and that the whole observation led me to biofilm research.
And so how important do you feel biofilm is to the assessment and diagnosis of chronic infections such as Lyme? Do you feel that the presence of biofilm hinders an adequate assessment? And can you even tell us a little bit about what is biofilm and what role does it play in sort of the life and survival of these organisms?
Those organisms, they want to survive, and actually they have a good head start. Now we know Borrelia is on this planet more than 50 million years. So they really had time to figure out what kind of strategies they can develop to not just to evade or antibiotics, but any kind of insult they might encounter in the environment. So biofilm is just one of the ways they try to hide from us. And everybody knows biofilm because everybody has a shower, which is almost, I always call a biofilm is like a city. So think about a city which is surrounded with all kind of protective layers. And you started to break down those layers, but you don't know what is coming out from it. What kind of different forms might come from these layers, and what kind of antigenic properties might have those forms. Borrelia changing antigens on the surface like we're changing on the rest. This is mind-blowing. How much insult obviously that the immune system can encounter when you have an organism which can put up a different antigen by daily.
Right. And what parts of the immune system are typically responding to the biofilm in these organisms? In your lab, do you see, is it the innate immune system with certain genomic markers that are becoming activated, or is it more so T cells and B cells and the acquired side of the immune system? What have you seen in your lab and how the immune system behaves?
Yeah, I'm not studying that part of the research. Right now, actually, what I'm studying is more like the interaction of Borrelia. Hello? Are you here?
Yes. Yep.
Oh,
yep, I'm here.
Yeah. I'm not studying the Borrelia and immune system interaction. I'm more studying Borrelia and the surrounding environment when it try to settle in the tissue. So right now, my main research is more focused on fibroblasts and mesenchymal cells and Borrelia, and we're finding very, very interesting observations. For example, we now understand that they interact with each other, like probably you heard about the cancer cells interact with the surrounding fibroblasts. We're seeing something very, very similar, that it is changing happening between Borrelia and the surrounding environment. And it's not, I'm not even talking about the immune system. It's just for example, fibroblast cells.
Right.
Connective tissue.
And Dr. Sapi, we were speaking before we started the formal interview, and you had mentioned that you often get the question, why is biofilm and in particular, why are chronic infections like Lyme becoming more pathological? Is it better? Is it just that we have better diagnostic tools and are beginning to understand how Borrelia behaves and the fact that it's maybe contributing to a larger burden of illness in the population? But you had made an interesting comment about our lifestyle. Can you tell me a little bit more about that thought?
This is, I live in Connecticut, so obviously I'm actually very close to Lyme, Connecticut, like 20 minutes or so. And I have lots of friends, and they come to me and they ask me, I don't understand what is this new number about Lyme disease patients. It's now 300,000 patients a year. And I said, what happened in 30 years ago? We had ticks in the woods. I remember, like one of my friends said, I remember picking up ticks from my body all the time when I was a kid. I never get Lyme disease. So what happened in 1976? And what I'm seeing when I'm meeting a lot of patients, that the first question usually, what happened the time you get diagnosed? And it was always something extremely stressful happened in their life. And that's what I try to see more and more trend, that is not just Borrelia, not just the tick bite, but actual lifestyle change in the last 30 years. And actually exactly correspond the time when Lyme disease rate went up very, very significantly. Obviously, the environment is now toxic, obviously, or food supply is not as nutritious as it was even 30 years ago, and our lifestyle is extremely stressful. So I think it's putting everything together. I think it's more us than Borrelia.